Hello, welcome everyone. Once again for the today we're going to start chapter 3, human reproduction. Chapter 1 to chapter 2 chapter 1 it's all about the introduction or the concept of or reproduction in organisms, right? And then chapter 2 we studied about the sexual reproduction in flowering plants. And then chapter 3 human samo. Hey, chapter 3 pin zoology part ito first and second mga botany part mo. As actually human lam to kisite zoology part hidin chin. Plants lam to kisite botany part. Like biology it comprises of both botany and zoology. So zoology part in chin. Reproduction to chapter 1 again sama banga. It is a biological process where mga organisms they give rise to offsprings grows, mature, and in turns produce offsprings in any organisms. And then now specifically we are going to talk about human reproduction. Mihing tiya ka bang tinando reproduction what are the reproductive organs, right? And then after that, why reproductive organs detail structure, functions because what is a chapter 1 what is gametogenesis? Gametogenesis is like the umbrella term. Gametogenesis the formation of gametes. So that formation of gametes, what do we call here in this human reproduction? Formation of sperms, pasala, formation of ovum or egg. So that two processes also we're going to study here, and then their reproductive organs. Yeah, so let's jump straight into the male reproductive system. We'll talk about the structure, the functions, how are the sperms formed, what is the name of the process of the formation of sperms, a process and then how what cell division is used there, and then what kind of cell division takes place there, and then after that we'll go to female reproductive organs, and then the process of how the egg is formed, why they have important, and then that will be it. So first of all, male reproductive system, it comprises of what is the primary sex organ and what are the other organs which are helping in the formation of the sperms. A primary sperm ki form na primary it helps, like it is like a helping hand, so we call them accessory. So we can divide in male and female primary sex organ primary team so a first pin, and then abana apan pituti bang accessory. Accessories kong mo ibang accessories kong le ibut mo le heat what's they were bag and all belt. They were kind of accessories to enhance the looks, right? So accessory it helps in the reproduction. So male reproductive organ will have primary and accessory organs. So a primary pin bang dia naging tao koy lag mun de hidiam le sperms ki form na pin testes say this one everyone knows testes in testes bang what is the structure present inside, then we have to study it, right? So testes banga box compartment bang sa oma bang chiki forma. So first of all, I'm going to divide into what is the primary sex organ, testes, and accessory hay vagian mo accessory akidot le chu hay gen primary kichile hay china accessory organs. It includes accessory ducts till him bio ducts in any chapter ducts tube tubes bang ducts is a tubes bang pipe pipe long asunga tuilwang the so till khat ki carry it can be blood, it can be hormones, right? So they are hormones till khat carry the white tube pen white gooey pen ducts ki we have also glands which will help, accessory glands. and apola ma imuk thei pen kha external genitalia ha chan to ki divide chu so hai tingle atoma zildi so hai pena ki focus pol lama testis pen kha banga oma sunga compartment bangzom. What are the hormones responsible for the formation of the sperms? Very important. This is all about hormonal control. If your hormones were imbalanced or if your hormones were not in the exact right amount or quantity, then there will be a problem in the formation of sperms. All right. As a hormones pen ha very important. Bang din naging tao le hormones pen. Testis nga hormones pen banga le. It's called the male sex hormone is known as what? Testosterone. Mala. Ahiya. Hormone pen bang. Hormone which will regulate, it is known as what? Testosterone. Kum somle ni puberty hit jang aha, boys and girls. What happens is that formation of a sperm pen hamale, or pen and formation of ovum pen hon ki panta it started. So testosterone hormones were produced in a large amounts. All right, when you hit puberty 11, 12, it means like you are now reproduction ha theta digina mo like coitus somle tani theta digina. So why stage pen ha puberty ha on hit jang ha. So the testosterone hormone, which will be obviously signaled by the brain, the pituitary, the gonadotropin hormones and anterior pituitary gland by the brain, and then that signal will come, the hormones will come through your bloodstream and they'll go to your testes and they'll say yes, now you produce testosterone, and then that is how we form the sperms. All right, so don't forget testosterone, male sex hormone, which will help in formation of sperms inside the testes. So now let us see the structure of testes, all right, and then we'll come to the accessory organs. Testes. Koya ki situado. What is the location of testis? Location. Where does it occur? Where is the location? Abdominal cavity. Mo leha igilnoy abdominal cavity to ki kusunzome. Mo leha ya. So abdominal cavity to kisonzomen sin why testis pen kha nyam suga omeju skin pen to chu kizomai wanga, but it is not attached inside, it is outside. All right, so within a pouch called scrotum, testis pen kha asunga ay apola ma kha akimu pen kha bang scrotum. So testis will have, it is covered by a cover to pen kha bang scrotum. Scrotum. It is a pouch, all right. It is a pouch-like structure. So scrotum in cover in chin. At sile hai pen ka scrotum ha asunga testis pen jo. It is inside, all right. It is inside. So scrotum ha apola ma pen in chin. Scrotum pen ha. As I told you, it is located, down and outside the skin. Skin pola ma ha to amangom. Jo bang zagaw ma why it's not like connected and then inside the skin or the abdomen. Why is that? Because a human normal temperature size, 37 degrees Celsius, right? Our normal temperature is 37 degrees Celsius, but the formation of sperms, which the process is known as spermatogenesis, which we'll study later, why formation of sperms, it cannot occur in the 37 degrees Celsius. It has to be lower. How much lower? 2 to 2.5. Don't forget. Keep in your mind 2 to 2.5 degrees Celsius are lower than the normal temperature. Then all this process, spermatogenesis, can occur in that lower temperature. All right. So scrotum pinha bangza outside the oma chile bangza, because for spermatogenesis to occur, it has to occur in lower temperature, that is 2 to 2.5 degrees Celsius lesser than the normal temperature. All right. So scrotum pinha 2 to 2.5 degrees Celsius lower than the normal human temperature. That is 37 degrees Celsius. So in that way, so that why? Because spermatogenesis can occur. Mala. Why? Spermatogenesis can occur. spermatogenesis is simply the formation of sperms. As actual 37-degree so a little bit of cooler, lower calm temperature they needed. And then that is the outside. Mola a skin penha, skin penha, scrotum penha, pouch-like structure. To asunga lu late testis som tadejo asunga ha. Alum atang mo, atang penha testis penha, entale bang jiran lewa ki formale scrotum penha. If you want to write down, you can write down, And then here I'm going with the second one, which is known as testicular lobules. Testicular lobules, B-U-L-E-S. I think it's better if we testicular lobules Alright, I'll write first and then I'll draw later. Cool? So it has lobules, testis compartment or major compartment. Compartment is like we have rooms, hostels with a compartment. So likewise they have compartments, and inside the testis they have around 250 compartments. Alright. And next scrotum theta. Scrotum theta asunga testis sunga bangom testis ka compartments are divide 250 250 home 250 divide in chin. Abane hai testicular lobules hai compartments sunga bangom. Inside these testiculars, what do we have then? We have seminiferous tubules. The name. Welcome to the name. This is so important. You have to remember it. Seminiferous tubules. Seminiferous tubules. Seminiferous tubules. This is basically where the formation of sperms takes place, right? So there will be so many cells involved and then so many hormones involved, right? So in this tubules, tubules. What is the meaning of tubules? Tubules, tube. What tube-like structure? More ke coil ja. Tamatam ke coil engine. Why sunga ha? Cells. Why cells? Why cells te ha? Banga. Seminiferous tubules sunga ha. Let's go to the third. That is known as what? Seminiferous tubules. Seminiferous tubules saha we have Sertoli cells. Kachibanga hai tube mo. Testis sunga tube ta matam why sunga ha cells or major cells. Changa cells mo cells. The first one is known as Sertoli cells. Sertoli cells banga a function thing. It has to do something with the sperms. Everybody has to do with something, the development or the giving the food, or it has to do with something, or the hormones, right? Because this is about the testis. So without the hormone, then they will not be able to produce the sperms. Hormone la poimo, an la poimo, hay te cells omten ha apiago echo. So Sertoli cells to banga boy chile amauha in nars janga mo dam doi hon pete dan dewa. So Sertoli cells or nars cells provides nourishment to the developing sperm. Sperm, like we'll see it later, okay? Sperm. So sperms are coming from the sperm mother cell. We'll see it later. A sperm ana mature saktu ana pitu pinha a cell som pinha bangay chile nourishment of developing sperms. Pinha Sertoli cells. Don't forget. And then we have, what is that? This is the one, how sperms are formed. We have one cell known as spermatogonia, or the germ cells. Germ cells is yung sex cells mo. Spermatogonia pinha, we call it male germ cells. Bangzera germ cells because they're the sex cells. Sex cells, why? Because they're going to give gametes. Alright, gametes bang sperm. So male gamete is the sperm in humans. If you remember, flowering plants, what is the male gamete? Pollen grain. Male gamete has sperm. So this one, spermatogonia, what happens is that they will form sperms. Clear, no? This will be all our cells were diploid, so 2N and sperm is haploid. This N. 2N apa N what kind of cell division we're going to use? Meiosis. So we have spermatogonia, number 1, Sertoli cells, number 2, spermatogonia. Abana outside has seminiferous tubules. A reason outside the seminiferous tubules we have one cell known as Leydig cells. Leydig cells ka interstitial cells. The other name is known as interstitial cells. What do? They are the ones which produce the testosterone hormone whenever it is needed or required for the formation of the sperms. So this one is testosterone. It produces testosterone. Yeah, you know, like tom thei pen amo tam thei lo di ai bang a tom thei de bang a kha hoi de wa thei chan de mo banga om. So testis sunga bangom scrotum pen kha normal temperature to kitwang lo chang kha 2 to 2.5 lower om thei di abana. It is a pouch like, and we have the inside we have compartments, and those compartments mole testicular lobules kichi. Why testicular lobules compartments sunga bangom the seminiferous tubules why sunga cells chithum chini it's why Sertoli cells which will provide nourishment to the developing sperms. Where are the sperms coming from? Spermatogonia kichi male germ cells apadaha sperms on ki form olola why in ki form na penaha anpetu pen bang Sertoli cells. And then outside the seminiferous tubules, this is outside, don't forget. Outside we have Leydig cells which will produce testosterone. Testosterone hormone asunga alu changa ha, you know, the process can start, you know, spermatogenesis and all can start. Plus testosterone is responsible for secondary sexual characters mole. Mulpo na hiya o tuam na chihong. Koa chi numetia ha estrogen mole. Olola kamole yote numetia high pitch pasaltia op you know puberty hit changa that sexual characters, secondary sexual characters is also done by testosterone. So let us read the diagram. Mole what again ting ha diagram ki draw le jang tadi to ha or so now draw the diagram of male reproductive system. Okay, human male reproductive system. Testis ay pa male direct gina. Structure of testis. Dun, for example, kachima bang let's draw big one. Testis mo. Scrotum pen so draw myself sila ka scrotum ita made mo la. Scrotum in a cover ha mo. Scrotum is the cover. Testis. Alright. The compartment kachiboy mo. So we have around how many compartments? 250 compartments, right? We have so many compartments. Li ho to draw him ni. Compartments ka omta. Aba compartments zo abang kachi. Compartments zo abang kachi. We have scrotum and we have compartments, and after the compartments inside we have what? Seminiferous tubules, right? So we have so many seminiferous tubules, which are made of so many cells. Let's see. Malaya. Tube-like structure, seminiferous tubules. As a whole, seminiferous tubules. I seminiferous tubules ka, let's say, inside, right? Seminiferous tubules. Two seminiferous tubules ka. Seminiferous tubules bang omkati. What are the cells to form the sperms? What are the cells? Cells bang sa chini omale. Why cells ke bang? Sertoli cells, right? So let's say this is the Sertoli cells. Sertoli cells Sertoli cells will provide nourishment to the developing sperms. And then we have spermatogonia, which will give rise to the sperms. So all the cells, there will be spermatogonia. Say these are the spermatogonia. Spermatogonia, abana, it will give rise to later anapiak janga ka asunga padaka sperms omdi. Ahiya sperms de omdi. Italian sperms de. Sperms de omda. Just spermatogonia will give rise to sperms by meiosis, obviously cell division. and for that to develop spermatogonia cell here. Cell pen sperm aswag theding they have to have food, and that food is coming from Sertoli cells. So all cells pen sperm. So sperm it has a head, it has a tail. Alright, we'll study the structure of the sperm in detail later. About Leydig cells apolama mention. Let's say here there are some cells outside. Oh my goodness, the red color is not working. Cells new apolama omchini. As also textbook mial lo mo ka like thesiam high load so haya panin hoi den leo jin mo thesiam so di Leydig cells. Leydig cells in khabang boldi testosterone produced dimole. So outside the seminiferous tubules, we have the Leydig cells. Leydig cells in kha, and here Leydig cells in kha bang chidi, they will give the hormone inside. Male hormone alujang kha, the process of spermatogonia into sperms can occur. Why kha a process kabang spermatogenesis? Why kha den lam just simdi. It goes the hormone goes. See the hormone when it goes inside, then what happens is that spermatogonia will eat the food and change its structure and shape, and they will form the sperms. So with the help of who? Sertoli cells. So what are Sertoli cells? Nerve cells. Clear now? This is the testis. What is going on inside the testis? When the boy hits puberty, and then and then it will come out of the penis, right? So how will it go? How will it come up and reach outside the penis so that it can be ejaculated out? So now here there is the problem. I have to erase this. You have to know who is which. Now let's see. Sperms dekha on pai tau jo ayaha kimukhom tau. Let's see here. Hey structure ha kimukhom na kan ngom de, and then they will come out. This is a scrotum, then they will come out. All right. Let's see. They will come out. That's just sperm. So next is now testis is over. Let us say the testis part is over. Let us see the accessory ducts. Accessory ducts is young tubes which will help in transfer of the sperms. All right. So sperms they have sperms they hon port kheta in high structure hon ki they will meet here, so that this is like a network of tubules. All right. We call it reed testis. Reed testis. The reed testis angkim taucho. Bana paito de unjin. This is highly coiled, okay? Highly coiled. Inside there are so many coiled tubules. So highly coiled. So highly coiled structure penha a second penha. Epididymis. Epididymis. Epididymis in temporarily it can store, okay? Store and also obviously the main thing is to transport the sperms. Why a store to pen kwa chile epididymis le di mo answer. Epididymis happen paito in then we have this one which is totally outside the testis already. That is known as vas deferens. Outside of Benmon. So here read testis, Avana. Hai akipat na hai abul pen le hai le omlai. Hai. Hai pen pai the chumo. Hai mun. Vasa efferens ya hai akipat na bul pen ha. Hai kha vasa efferens ya hilai. Vasa efferens ya. That's examine tom ni why because of this location because they are all this tube-like structures, highly coiled structures which carry the transport the sperms. They are ducts they hai sumtana hai ki mukhom na kha reed testis kichi hai koi namuna kha vasa efferens, and then when it comes out here we call it that long tube. We call it epididymis, which store and transport the sperms and then go totally out of the testis. achu ureters lama hon pai to ta hoi pen kha vas difference pen kha alas pen to sperm hon pai ta vas difference kha ta this sperm which is carried, what will happen is that accessory duct stage. Are we clear with the accessory ducts? What are the accessory ducts? Come on, number 1, reed testis. Number 2, vasa efferentia. Number 3, epididymis. Number 4, vas deferens. Male, these are the accessory ducts. Testis, primary sex organ. And the sperm has to be carried through the ducts. And sperm has to be mixed with some, some liquids, mga tui the hon ki mix ngai like this. So to be mixed with that liquid, Chimeni, why pen ha, koya panang ki pandai chile glands. That is where the accessory glands come into play. Accessory glands te koi omode. Why on porke tansin, why ha pasaltia urinary bladder mod zunbo. No ya ha alang ha. Kaya vas difference by suna ha structure ni appear nom suksu. Why can ha hon contribute? They'll contribute something of their own, and then it will mix up with the sperm, and then it will come out as semen. So what are those things? Let us see. First, we have seminal vesicle. Urinary bladder here. Urinary bladder is here. But we are not, but it is the same thing, common part with the, urine. So urinary bladder will be here. But we are talking about, the sperm. So here, right, there are two structures, which is known as seminal vesicle. We are talking about the glands now. Seminal vesicle. Seminal vesicle. Bana. So now this vasa efferentia. Efferentia. Ayan ay lamagyan minimo. So seminal vesicle. Has seminal vesicle in height will contribute something. What does they contribute? They contribute. We call it fructose. Sugar mo na. Fructose. Bana calcium. Yeah. Calcium and so many enzymes. Enzymes tampili pedio. Fructose, calcium, enzymes. They contribute, all right. And then we go to the next prostate gland. We have next prostate gland, same thing. Prostate gland in le bangdi. I palpi thodi mo I tom te all these vitamins and all those prostate gland. Prostate gland. Can khate pasal te if there is some banga boy na khong new chang ale prostate ale cancer ong themo hai ha prostate cancer le kichi. Prostate gland. Abana next aha bulbourethral gland. We have another one. I pen khabang bulbourethral THR gland. Gland ngan ka chiboy mo mau kha gland sin gland si changa de secrete mo secretion secrete uchi nai chu. So bulbourethral gland pen kha lubrication. Anal na di sperm te move fast hai de lubrication ha. Lubricating gland ka chimo lubrication, am I right? For lubrication. It's a pang khomo changa ha sperm te khai hai te ton ki mixta fructose enzymes mo le hon tui tai chu mo le, and then it come out as what? Semen. Here, why semen pen kha? It will be milky whitish in color. Why semen up in ha sperm suit? Obviously it will be there. Male sperm suit contained. Kilele why on ki ejaculate changa ha why pen kha semen kichi e pa bang bo tui ai kha bo tui bomb mo a bomb status ka bo tui bomb kichi. So now we get male gamete, that is a sperm, all right, and then female ha XII, and then you know that after that fertilization a process ke pai. So now I have to really understand, how it goes. Testis are compartments, more than 250 compartments, and we have what? Seminiferous tubules. Don't forget that is a place. Seminiferous tubules, where the formation of sperms are. Seminiferous tubules are, we have two cells, Sertoli cells, which will provide nourishment to the developing sperms. And then where does the developing sperms come from? Spermatogonia. So the next is spermatogonia, male germ cells. Male germ cells. We can also call, like, you know, stem cells. Ki form themeu, you know, lemon sila ki hormone mo, testosterone hormone they can form again and again. So we can also call stem cells, right? So why spermatogonia tampi ki form why penha can put zelo abana ki form to zelo ma. So spermatogonia penha sperm on swaga here by spermatogenesis process. They undergo cell division, meiosis. Male, 2 and penha and on swag because sperm is always haploid. Spermatogonia ha diploid. Clear? Aba na khwai ha aki transform ding ha Sertoli cells in panpi as a nurse. Male. And then it comes out and form here. Hey, compartments there. Hon kimuto anapin ha bang reed testis. Reed testis apada ha ya ha hon kai to na penha bang vasa efferentia. And then tunga saw penha highly coiled penha epididymis that store and transport sperms. And then testis polla maom outside. Polla mahon chang habang kichi vast difference. And then a sperm penhang tai sup in genaba na secretions will occur from the seminal vesicle, fructose calcium enzymes, mole, peramu, prostate gland, number 2, these are the glands, and bulbourethral. Plus, all this, plus, it also helps in what? Lubrication. And it comes out as a white milky color, which is known as semen, so semen will be rich in what? Fructose, calcium, enzymes, mole, toma. So, here tingom tinitinga ha, koya para kipan, banga hormone, testosterone. This is so important. And testosterone, where does it come? How does it produce? Here para testosterone bangchalanda waluda. Testosterone hakwan haluta chia. When it hits puberty, your brain signals gonadotropin hormones, then it signals pituitary gland, and then it comes out through the bloodstream. It goes and travels, and it goes to your testes. Male, lutawa testosterone na se patetachi. So why testosterone lungtang like stress brain la healthy lo wale testosterone le hoy ke produce lo the here. So it's all connected, you know, the physiological thing, hormones dekha. So hai teng chau healthy the hilo na lungtang apa da healthy di hon ki signal the di signal chang testosterone hoy de watam di atam chang hai formation om the pande. So if you have like problems some lekha testosterone hormones imbalance the ma. So testosterone hormone imbalance there are so many problems. Male, sperm ti dam low healthy low ma. Chi khong hilo le banga erection by tail chi khong problem tam. So you have to eat lots of nutrients, vitamins, proteins, so that you will be healthy. Male, in the fertility di ma. So basically semen penha, it will be your fructose calcium penha waiha bangkichia. Fructose calcium enzymes together we call seminal plasma. What do we call? Seminal plasma. No. Semen contains seminal plasma and your sperm. And then now we will see why spermatogonia was sperm former. I'm talking about the cell division. So we'll see that one. So how is sperm formed? Alright, the process is known as spermatogenesis. Spermatogenesis, formation of sperms. Mole. Formation of sperms from the germ cells, it is known as spermatogonia. Spermatogonia, a germ cell. Where does it happen? Inside the seminiferous tubules. Male. Kaya oma seminiferous tubules mahilo dio compartment again lo di seminiferous tubules ngahi ay cells tang omna spermatogonia omna sertoli omna. Eva Leydig cells to outside here, don't forget Leydig cells to outside. So the formation of sperms for spermatogonia, that is the germ cells or the stem cells inside the seminiferous tubules by the process of meiosis, is known as what? Spermatogenesis. So why seminiferous tubules are cells so may puberty amo. And then what I forgot to tell you is that sperm ka hon pot chang amo khat ve thuan pot chang ki ejaculate chang bangsa ki ejaculate so bangsa din gi taw khat ve sperm apola ma pot chang ka bangsa din gi taw 200 to 300. Khat ve thuan mo 200 to 300 ko ite. Hai tinglaga khat cho hilai cha obama lut zorni pen khamale fertilization ki gen zauta anasada. So spermatogenesis ka spermatogonia ma hai spermatogonia hita. This is the mother cell which will give rise to the sperm. So spermato kipanaha a mother cell pen spermatogonia. Diploid mo haploid diploid male. I told you it's the diploid, right? Diploid it's a bang. A chromosome number bangza 46. Hapen kha bangza itung sagdiu 23. Mekha n. Yeah. So that is why we need what kind of division? Meiosis. Clear. So spermatogonia in kha. They'll form what? Sperms, which is haploid. Male. And to form these, we have nerve cells, na? Remember? Sertoli cells to develop into the head and tail sperm, where this is just a cell. So spermatogonia what are this? You know, two cells in between in this development. Spermatogonia, they differentiate it jang khat chaw gilawi jang mo ta matamun jin white ta matam te kha hon ki differentiate ta matamun so with jen abasa pian kha we call it primary spermatocytes. A form site jang kha primary spermatocytes. Primary spermatocytes ha still twin. Zakchu he kha meiosis of nailo ya mitosis ipot ada chu mau ki differentiate me mo he kha the siap di atak tak pian nailo. So mitosis is one will become two will become four more. So they're just mitosis here first because this is this primary spermatocyte. So it will be still 46. Now the real thing happened. Meiosis 1, meiosis anomta. Meiosis anomta this one will become a second form secondary spermatocytes. Meiosis of a chromosome number habang hisa n, right? Good. So we'll have secondary spermatocytes. Two secondary spermatocytes it will undergo another meiosis. Meiosis 2 hita di mole one khazauta here it will be meiosis 2. You know that meiosis 2 equational division of mitosis to kibang mole ka chi changa chu meiosis changa hai n bana 23 bana half a reduce non long di kibang tadi 23 hita di mom that's equational changa chu mitosis equational division reductional to chromosome reduce meiosis, right? So in meiosis 2 it is like equivalent to mitosis, so there will be no more reduction in the chromosome number. Okay, don't forget n here to 23 might hidden then the sperm hitanamo. So haya ha aki form pen ha spermatids. Spermatids ka alu hong banga a structure a shape hong ka fukchat nai lo chuneni, so they'll differentiate and they'll form sperms. High differentiation pen ha shape and structure will change. Size. High shape, structure, size change na pen ha hai pen ha bang kichi. Hai only this part, you know, it is known as sperma, mga sperma. Spermatids para spermatozoa. We also call it spermatozoa. I think of spermiogenesis. Yes, spermiogenesis. I think spermatogenesis. Otherwise, the development of the sperms from the spermatogonia, this whole thing is spermatogenesis. Only this differentiation in shape, size, that head and tail more sperm. So that only that part is known as spermiogenesis. So what is the difference between these two? So let's say how the meiosis occurs. So by meiosis, the 46 chromosome becomes 23. Why it has to become 23? Because this sperm has to be, fertilized with egg. Egg is also 23. So 23, I can mix with 46. So I can human digzel male. And then here process test this pasel 46 para 23 ki reduce deal. Numediale 46 para 23 na reduce di fertilization on 46 on his LD ka naungi ka mo mihinga 46 his L abana iale it keeps on going and going. This is just maintaining the constancy of the human chromosome number, right? We have 46, right? 23 pairs. So why done my age 46 to 23 ke kalmo. So meiosis wala the real reduction takes place here from 46 to 23. Meiosis to equational time, so it will be same number. So I draw that. If I draw, I think you'll be able to understand. For example, I have spermatogonia. Yeah, spermatogonia. Mitosis just differentiate and they form, let's say, two. They form so many. One will form two. One will form three. One cell will form two. Likewise, others will form their own two. I be cut to any lie because mitosis. Meiosis one so 2 and pen habang itali and on tayo here and 23 tayo mala 23 ta secondary spermatocytes hapat 23 ta, don't forget. And I have 46 chromosome 23 ta 23 to I have 46 to sila spermatids ben ha 23 apa half ki reduce non lo de ka chewe male azaju why meiosis 2 is equivalent to mitosis male. Meiosis 2 ele in class 11 na boy wala mo ki bang equation of ki bang, so there will be no more trains. So spermatids ni maundi, so it will be like mitosis ka chewe mo equation na like divide with chang ha to on swakta yeah spermatids ka li on swakta because meiosis ka it is equivalent to equational division of mitosis. Equational division of mitosis. Equational division of mitosis. So ki bang tay chang ha. Habana tuhon swakta. Habana hekha n hitamo le n don't have to further reduce it male. So this one, what will happen? Certainly cells that unpure, so they become structure, shape, size, extension, head on later, sperm on swakta. Oh yeah, we have 4 sperms. All right. So one cell form 4 sperms. We have 1,000 of cells. They'll form 1,000 of sperms. We have a million of we'll have millions of sperms, and that is how 200 to 300 million sperms is ejaculated at once. it's done the spermatogenesis. So once spermatogenesis a sperm bang zaki produce for bang chidan dewa as chu spermatogonia juwen. They form primary spermatocytes still juwen ma secondary spermatocytes reduce because there goes the meiosis. So 23 hita about na hetele abona 23 hita. As actually this is like 23 hita. And then high 23 human abang chen no constant ka chu hai sperm kha 23 ta chumale chromosome number na egla 23 chumale. How is it reduced? You'll see. And we have what? Zygote khabang 46. And then like from here zygote mihingai changa mihing khadele nume hile like they'll have ovary and pasal hile testis neidu testis sunga spermatogonia 46 apada 23 hon swag zeldi nume diale male ogonia apada XII hon swag zeldi 23 and then it goes again 46 divided into it will reduce to 23 and life goes on. Mainly more again the non ke tidan the wage. It has a head, simply more, simple to draw the I have a labeling a dignity. A head pin in her, a chromosome. A chromosome pin contains, chemicals in her, over aluchanga put in a dingwa, a heart pin, put the changa chemicals in her, a ovum, a layers de hamo, put in Genesunga, XII pin her, of a lutenadingwa enzymes, a secret now. A secret division, and then it will be open over layer pin. Hamo why is a chromosome very important because it contains enzymes so that it will push or it will break what the coronal radiata. Structure of ovum and leucina coronal radiata to sungaha bang pe zona pellucida hilom zona pellucida tiaka enter of ovum. So therefore they need a chromosome. It's contain lots of enzymes. So this is the head. This is the neck. This is the head. Atel pencha. Ovum atluchang atel pencha kinusia mo. Atel pen poimolo. Atel pen poimo nachu. Swimming di jawa. A middle piece. A middle piece aha. Athani ting tai the ding waha energy pitu pin hayom energy ko yaparan kipan mitochondria to energy currency of the cell. Not all the cells will have nucleus or all this so they have mitochondria ka energy pitu energy currency of the cell like jima for swimming wala motility kichi motile sperms are motile, they can swim. Ovum they cannot, so they just wait for the sperm to come. Energy source for swimming. Structure of the sperm, simple penama. Last but not the least, I want you to talk about the hormone. Alright, first brain gonadotropin hormones, releasing hormones generate mecho. Gonads reproductive organs gonads gonad more reproductive hormones. Brain anterior pituitary gland. Yeah, signal peta. Why hontai sukin chin testis on lucha. Testis hormone on secret. Why hormone te khabanga? No mayale pasalale kibang luteinizing hormone. Luteinizing hormone in Leydig cells. Yeah. Now we can secrete what? Testosterone. So they started testosterone. They are present outside the seminal vegetables. Alright. Follicle stimulating hormone in follicles Sertoli cells. Sertoli cells. On forms against nerves engine and engine so that the sperms will mature, right? Maturation of sperms. Then they will come out through the root testis, vasa efferentia, more epididymis and vas deferens, and then the seminal, vesicle, bulboid glands, postjet glands, they will produce their glands. The glands, I mean like enzymes, fructose, calcium. So it comes out finally as semen, which contains the seminal plasma and the sperm, and that is how it is done. The process of spermatogenesis, and these are the hormones involved. Brain lamto reproductive organs like no one is working on their own, everything is like teamwork. Next we'll see the female reproductive system.